Hello everyone, welcome to another episode of Anthropology lecture series. Today we'll look into polygenic inheritance. This was a question asked in UPSC Civil Services Mains 2023 examination. So what is polygenic inheritance? When we see the term inheritance, what first comes to our mind is Gregor-Mendel and his monogenic inheritance. But human traits are much more complex. Simple monogenic inheritance cannot fully explain how human traits are expressed. So that's why we have polygenic inheritance. So let's see what polygenic inheritance is. So let's break the term polygenic. Poly and genic. Poly is many. Genic is something related to gene. So we know for one gene, there are two alleles. While if there are poly means there is more than one. So when there are two genes, there are four alleles. When it comes to three genes, six alleles. So more the number of genes, double the number of alleles. In polygenic inheritance, all these genes have some role. All these alleles have a cumulative effect on our trait expression. So this is called polygenic inheritance. So R.A. Fisher gave multiple factor hypothesis. R.A. Fisher gave the theory of multiple factor hypothesis. This hypothesis is that for human trait inheritance, there are multiple factors, not just one factor. So the first factor is the cumulative effect of all the genes and alleles. And the second is the environmental factors. So combination of environmental factor and the cumulative effect of genes play a role in human trait. Let's take an example. Our human height is controlled by polygenic inheritance. So a trait called height is controlled by more than one genes. And all these genes play role in determining a person's height. How tall a person is determined by a number of genes and the environmental factors. So now we'll see the gene and allele. Here, this is gene A, B, C, and there are two alleles, AA, BB, CC. As I told you earlier, more the number of genes, double the number of alleles. Now we'll see the characteristics of polygenic inheritance. The first feature is incomplete dominance. We all know what complete dominance is. In Gregor-Mendel's complete dominance, if there is two alleles, one will be dominant and the other one would be recessive. Whereas in incomplete dominance, no allele will be dominant over the other. All will have a cumulative and a combined or a blended effect on our gene. Say for example, if we cross two homozygous parents, here this is a pure negroid, A, B, skin color, and this is a pure caucasoid, A, B. So if these homozygous parents are crossed, we'll get a heterozygous mulatto, and this skin color would be the intermediate or the blended form. This is the result. So this is what is called incomplete dominance. So the next feature is continuous variation. When it comes to monogenic inheritance of Gregor-Mendel, the variation is discontinuous. Say for example, our hairline. For some people, there will be a widow's peak. For others, the hairline would be straight. So here, this is a discontinuous variation. But when it comes to polygenic inheritance, like skin color, we can see there is extreme values, and from extreme values we can see a variation. No two people will have the same color. There is in light there is more light, in dark there is less dark. So that is called continuous variation. So now let's see another feature, that is the environmental effect in polygenic inheritance. Environment plays a crucial role in polygenic inheritance. Let's take the example of two diseases, diabetes and hypertension. Both diabetes and hypertension are hereditary in nature. Both can be controlled if we control our environment. So if we control our diet and do proper exercise, we can control both these diseases. So this shows how polygenic inheritance is impacted by our environment. Next feature is that from all these discussion, you might now understand that polygenic inheritance is much more complex than monogenic inheritance. So this is another feature of our polygenic inheritance, the complex nature of polygenic inheritance. Now let's look into another example where polygenic inheritance is shown by our human body, and that is our skin color. So this is a Punnett square that I've constructed here, and this value called 6 is pure black, AB, AB, ABC, ABC. And this value called 0 is pure white, small abc and small abc. And these are the intermediate values that we get from this Punnett square. Here we can see those with the extreme values, that is 0 and 6, are of the lowest frequency. There is just one value of 0 and one value of 6. But when it comes to the median or the intermediate colors, there are more number of individuals in the intermediate range, 3. So this Punnett square is plotted here as a graph. This is y-axis. Y-axis is the number of individuals in a population. Here we can see, since the frequency of pure white is less, the extreme cases, both here and here, are less, values are plotted, whereas the intermediate stage is a maximum. So once we plot this Punnett square as a graph, what we get is a bell graph. So from this we can see our earliest traits, that features of polygenic inheritance that we just mentioned, the continuous variation, and where the mean value is the highest and the extreme values are the lowest. So this was polygenic inheritance. Hope you liked this video. We'll see you again in another video. Thank you.